on to preclinical. So we're moving a little bit back in the research timeline. And I have Dr. Charly Lai, who's here from Indiana University, and she will give an overview on preclinical research. So let me make sure. Great.
Yeah. Can you see me and can you hear me?
Yep, we sure can. We can see your slides. You look great.
All right, cool. So, hi, thank you for having me. Good afternoon. Well, I don't know the time zone. So it's very, very exciting to me as well that this morning I heard a lot of cool things about promising clinical trials, and also learned those very cool new devices, and then a bit from Jim, like patient's perspective. So now I'm going to take you from clinical back to preclinical to animal models and then to cells to tell you how do researchers use cells and animal models for the study of pulmonary hypertension. So pulmonary hypertension, which is high blood pressure in the lungs. So normally this is the right heart, the left side heart, and this is the pulmonary arteries. So normally the wall of the pulmonary arteries is thin, so the right side of the heart can pump blood easily through this low pressure system. However, in pulmonary hypertension patients, the vessels of the pulmonary arterioles becomes a bit abnormal proliferated. This happens in like a small pulmonary arterioles, small vessels, as well as in medial size or larger size vessels. So it's everywhere. Plus the loss of small vessels and inflammatory cells infiltration, as well as this called the neonatal neointima formation, meaning that you have new vessels grow in the existing vessels. So it's a lot of abnormal proliferation of the cells in your vessels. So the vessel becomes thicker, more stiff, and then you can see the lumen becomes narrowed. So the blood from the right side of the heart becomes difficult to pump through the vessel. And then so in this case, the pulmonary vascular resistance goes up, and then pulmonary hypertension, pulmonary pressure goes up. And then also right side of the heart has to pump harder in terms of to pump blood through the vessels. So eventually, when over time the right side of the heart becomes bigger but weaker, and then eventually it becomes like right heart failure. So patients with pulmonary hypertension and right side heart failure usually present with fatigue, chest pain, like Jim mentioned earlier. And they also present with light headedness, as well as edema, complain about exertional dyspnea, meaning that they have shortness of breath during exercise. They also have exercise intolerance. So a lot of these symptoms are actually very commonly associated with different disease. So pulmonary hypertension is very difficult to diagnose. So usually physicians, through the physical exam, medical family history, blood test, echo, as Jen mentioned earlier, as well as X-ray, they can figure out some signs of pulmonary hypertension. However, the gold standard for PH diagnosis at the current is still right heart catheterization. Maybe in the future, cardiomen would be the best, but currently it's still right heart catheterization. And basically, as mentioned by Dr. Benza earlier, so the catheter is putting through from the neck and to your right side of the heart, and then put into the pulmonary artery to measure the pressure. So in the past, the pressure, normally the pressure is less than 25, but recently there's some debate, and then it is being revised to 20. So depends on the pressure, the degree of the pressure. You can be like diagnosed as mild pH, moderate pH, or severe pH. And then during the right heart catheterization, the catheter can actually be further advanced into the pulmonary capillary wedge position, and then that pressure can tell you if there's any other backward pressure from the left side of the heart to help with diagnose classify of the pulmonary hypertension group. So as mentioned, that there are 5 different types of pulmonary hypertension. Group 1 is associated with pulmonary arterial abnormal proliferation and pulmonary arterial hypertension. It can be idiopathic, it can be familial, it can be drug or toxin induced. Group 2 is associated with left side heart disease. Group 3 is associated with some underlying lung disease or hypoxia, which is low oxygen. Group 4 is related with chronic blood clots in the lungs. And then group 5 can be some secondary pulmonary hypertension associated with secondary to some unknown causes. And as you can see here, so group 1 pulmonary arterial hypertension and group 4 CTEPH, they actually have some treatment options available at this time point. However, there are still no treatment for group 2, group 3, and group 5 pulmonary hypertension. So among these 5 different groups of pulmonary hypertension, the group 2 PH associated with left side heart disease is most common. As you can see here, the prevalence of group 2 PH is about 4,500 patients per million people, compared to 15 to 50 per million for group 1 PH. And today, I'm going to show you, use the diastolic dysfunction PH associated with left side heart diastolic function to show you an example how researchers use animal models and cells to study pulmonary hypertension. So the pH associated with diastolic dysfunction is now also referred as pulmonary hypertension associated with heart failure with preserved ejection fraction, PH-HFpEF. So basically, like Patty mentioned at the very beginning of the introduction, the heart, it can still pump very well, pump normally. However, the heart cannot relax well. So the pressure cannot flow properly into the heart, especially the left side of the heart. So because of this, like a plumbing issue. So when the toilet stock, the water coming out from the toilet. So the same thing happens to here. When the pressure, so when the feeding pressure goes up, the feeding pressure has to go to somewhere. And now the pressure goes back to the pulmonary circulation and then to the right side of the heart. So eventually, these patients, they still get the pulmonary hypertension and the right side heart failure. So because these patients with PH-HFpEF, they have both left and right side heart dysfunction and failure. So basically, they develop more severe symptoms than those with HFpEF and suffer significant exercise intolerance, frequent hospitalizations, right heart failure, and reduced survival. And PH-HFpEF is associated commonly with obesity, diabetes, and hypertension. As you can see here, the gray one is for PH, and then the black one is for PH-HFpEF patients. You can see that PH-HFpEF patients, they are more commonly associated with hypertension, obesity, diabetes, and hyperlipidemia compared to PAH patients. And more dramatically, more than 90% of patients with PH-HFpEF, they have been shown to be associated with 2 or more features of metabolic syndrome. As mentioned to you earlier, that there's currently no treatment for group 2 PH patients. And the worst is that the use of effective treatment approved for PAH in these patients with PH-HFpEF has been shown to be either ineffective or even harmful. So they don't have there's no treatment. And in the past, when I started to study PH-HFpEF, there's no animal model. So when we start doing the research in PH-HFpEF, we first try to develop a preclinical model for PH-HFpEF. Because PH-HFpEF is associated with metabolic syndrome, so here we use the rats with multiple features of metabolic syndrome. They were born with obesity, diabetes, and spontaneous hypertension. And then we inject sugin, which is a VEGF receptor 2 blockade. It is widely used in combination with hypoxia for the animal model to do the PAH, the group 1 PH study. So it is actually an anti-cancer drug. However, about 3 decades ago, the researchers, they unexpectedly found that this drug can induce endocellular injury in the pulmonary arteries. So we combined the sugin, which is single injection of the sugin in this rat with multiple features of metabolic syndrome. And after 14 weeks, we measure their pulmonary pressure. Because the rats and mouse, mice are tiny little, we cannot use the catheter to, we cannot put the catheter into the right position in their pulmonary arteries all the time. So here we use the pressure, we measure the pressure in the RV for the indication of pulmonary hypertension. So here is the RV pressure in the lean animal, and then this is RV pressure in the RV systolic pressure in the obese sugin animals. As you can see that the combination treatment of sugin in these animals, they have higher pulmonary pressure compared to the lean untreated animals, and their vessels become thicker. As I mentioned earlier to you, the vessel wall becomes thicker, and also they have bigger of the right side of the heart. And we also check their left side heart. They do have a left side heart, bigger left side heart. They also have diastolic dysfunction in their left side heart, and then they also have preserved ejection fraction. So pretty much this animal model recapitulates majority of the features that show in patients with PH-HFpEF. So using this clinical model, we tried to test different drugs in them. So far, we have tried 5 different drugs already. And today, I'm going to show you the first 2 drugs that we tried at the very beginning, which are nitrite, which can be metabolized to nitric oxide, which is a potent vasodilator. And nitric oxide has also been shown to reduce the proliferation of the smooth muscle cells. And then nitrite has also been shown to reduce PAH in animal models before, the sugin hypoxia model before. And another drug we tested was metformin, which is the first-line drug for the metabolic syndrome. So pretty much we give the drug in the drinking water to the animals for the whole time. So here is what we show, what we saw. From the RV systolic pressure data, we can see that the oral nitrite treatment, 2 different doses, as well as oral metformin treatment, they decrease the RV systolic pressure in the obese sugin animals, as you can compare their pressure to these untreated ones. And then from their vascular remodeling, we can also see the red color, we stand for smooth muscle actin, which is showing you the thickness of the vessel. If you can see the red portion, you can also appreciate that the treatment of nitrite and the metformin reduce the pulmonary vascular remodeling in these obese Suchin animals. So in patients with PH-HFpEF, recently the data was also shown that the inhalation of nitrite as well as nitric oxide, they decrease mean pH pressure in these in patients with PH-HFpEF, and this is in 10 patients with PH-HFpEF, and now the inhale it's into the phase 2 clinical trial. And to us, the basic scientists, this data is very promising and very encouraging, because this is a good example to show that animal models can help, and the findings in animal models can somehow be translated into the human disease. And now there are 2 clinical trials going on with nitrite and metformin. One is oral nitrite. This is actually a 22 weeks open label trial, and the 10 weeks data was published, not published, was presented at the American Heart Association meeting November last year. And actually, people from the presentation, we can also learn that the oral nitrite treatment decreased mean pH pressure in PH-HFpEF patients, which is very similar to the nitrite inhalation data. And metformin trial in PH-HFpEF is also going on, and both of the trials are being held in at the University of Pittsburgh at this point. So this is an example, and now I'm going to go deeper a little bit to show you the mechanism part. So basically, throughout the years of study, we try to understand what is the target or what can be a potential regulator in this PH-HFpEF. So we found that SIR3, which is a mitochondrial deacetylase in skeletal muscle, may be a key. So as mentioned, SIR3 is a mitochondrial deacetylase. Normally, its function is to detoxify this reactive oxygen species, which is a little bit like a harmful accident if you have too much, because it can damage the cells and DNA, RNA, proteins. So usually it's a harmful thing. And when mice don't express SIR3, it has been shown that they become older, they develop a metabolic syndrome, and pulmonary arterial hypertension. So in our hands, we found out that when we knock out SIR3 specifically in skeletal muscle, as you can see here, the SIR3 protein expression is decreased or is lost in the muscle, but they still express in the liver and in the lung. When mouse don't have SIR3 in their skeletal muscle, we can see their lung structures from CT scan. You can see that they have decreased the pulmonary vascular density, especially the small peripheral arteries. This is very similar to what I showed you earlier in the introduction. And also, the skeletal, when mouse don't have SIR3 in their skeletal muscle, they have thicker of the pulmonary artery. And this is also very similar to what I showed you earlier. And also they have a slightly increased pulmonary pressure. So this data suggests that there is a skeletal muscle and lung crosstalk in the regulation of pulmonary hypertension. So currently our lab is also interested in another protein called WWOX. So it is a tumor suppressor. It has been shown that in pulmonary arterial smooth muscle cells, in cells, if you don't have this WWOX protein, the smooth muscle cell proliferate more. And then if you knock out, if you delete wax protein in the mouse, in their smooth muscle cells in the mouse, they also develop pulmonary hypertension induced by hypoxia. So what we found is that the wax protein decreased in skeletal muscle, in PH smooth muscle cells isolated from skeletal muscle specific surgery knockout animals. So now we are trying to understand how surgery in the skeletal muscle, how surgery in skeletal muscle communicate with wax in the pulmonary artery, in the lungs. So we try to focus on myokine, which is protein or peptides that can be released out from muscle. So this is the direction of the study in the lab, and this is also an example to show you how cells and animals help in terms of pulmonary hypertension study. So in summary, I hope that these examples can show you how researchers use cells and animal models to study and to try to find treatments for PH-HFpEF or PH patients. And this is just an example which was summarized back in 2016, which is 4 years ago already, to show you that there's a lot of drugs or potential mechanisms that are underlying the pipeline of the drug development for the treatment of PAH. And finally, I would like to thank a lot of people behind the things. This is my old team at the University of Pittsburgh, and this is my new team at the Indiana University. And if anyone who's curious who I am, I'm here. This is me. And thank you for your attention. I would be happy to take questions.
That's fantastic. Dr. Lai, thank you so much for this great introduction to preclinical research. You know, one of the things as I was listening to your talk, and super fascinating science about pulmonary hypertension associated with HFpEF, is, you know, I was thinking, you know, if I were sitting in the audience, this is a good example of how we use animal models. You know, we talked a lot in the first section about all the stuff that we do in humans, but you gave us 2 examples where you went in and can make genetic alterations to the animals by shutting down genes, which we obviously can't do in humans. And in so doing, you can kind of cut certain things out of the pathway and see what happens. And lo and behold, you get pulmonary hypertension or other, you know, end organ manifestations, health problems associated with that. So that's just fascinating to me. And the other thing that I thought was really interesting was that you made an important point about how the animal, you saw some, we call it equipoise, when we see a certain signal in humans, like the nitrite human study. And there was like some data that showed that might be beneficial in patients, people that look kind of like the animal model. And how that's really important, because sometimes we come up with animal models and they don't necessarily pan out. Like, we start with a lot of stuff in science and they don't necessarily go fully towards getting to the drug treatment. So,
yes.
So you seem to approach this by also looking at data from humans as you're coming up with your model of disease.
Yeah, so actually this is a lot of team effort. So normally the basic researchers, the pen for basic researchers, that you do a lot of research in animals and you don't know whether that one day can be translated into human disease or not, because first of all, it's a lot of collaboration with clinical sites, right? So it your findings have to be exciting enough to trigger the clinicians that, oh yes, I want to do this in patients and this may be beneficial to them, and we will spend a lot of money and then try to do some researching then. So this is a lot of team effort, and I was very lucky to be involved in this basic science, basic research part, and then the clinical part. Of course, I did not do the clinical study, and to me, it's also very encouraging and that we have to do, as a basic researcher, we have to do right thing, and then report the right thing from the animals, because it may affect the patients. If this is not a good drug, then what if you give that to patients and then that will have some side effect that is not good, right? So well, again, I was very lucky to be involved in both sides, and then the data coming from the clinical trial, that is also very encouraging me to try more different drugs and different strategies in animal models, in hope that in the future we can try to help us, the society, the field.
Fantastic. I got a question here. In your animal models, did it matter the age of the rat or the age of the mouse that you used?
Good question. So we only tried that once. So when they are more aged, their pressure gets a bit higher, but that is not too much higher than the younger ones. Yeah. Mm-hmm. But in general, because aging, for example, PH-HFpEF, usually the patients have PH-HFpEF, they are more than 65 years old. And for PH, it's more from 20 something to 60 something or even older. So it's more broad age range. But surprisingly, we did not really see that in our animal model, aging. Probably it's not age enough when I use the aged animal. It's very difficult to define the age as well.
Fantastic. One more question. You mentioned in the model I think the mouse model, that one of the things that happens is they age more quickly. Can you just tell the audience what you mean by aging in lay people terms?
Okay, in lay people terms, so
how do you measure? How do you quantify that?
Actually, at the time, it was very striking to me when I went to these surgery meetings. I could see that they show you the picture of wild type and the surgery knockout animals. I don't know, because we usually see the animals, so we can see their look. First of all, they look just like aged people. They look older, and their activity is also, they also have less activity. So when they are in the cages, the wild type animals, they run around cages all the time, especially at night. It's when they are more active. But the surgery knockout mice, they just sit there without moving. So
fascinating. Wow. Well, thank you. Looking to see if there are any other. I don't see any other questions. So with that, Dr. Lai, thank you so much for this overview and for sharing your research with us.